Today, I'm here with Dr. Zachary Wong, Dr. Megan Milbrath, Dan Wines, and then you just heard from Katie Brandt. So we're going to just go ahead and jump in. Our programs are open to everyone. All right. I'm going to get us started with some announcements. So if you're looking for a resource that we mentioned tonight, try going to this website. I tried to compile all of the websites and links that we're sharing tonight to our video page for tonight's webinar. So you should find it through the QR code or the link, and that's where we'll share our resources. So a few announcements. One is that there is the annual beekeeping survey going on. It's through Auburn University and the Apiary Inspectors of America this year, and they are asking beekeepers to share their colony information from the late beekeeping year now through the end of the month. So it helps people out if you fill out that survey. We compile a lot of resources on our page, Getting Started with Beekeeping in Michigan. A few things to highlight. We have our list of upcoming events. It's a busy time of year for beekeepers, so we have a lot of events coming up. We have a list of workshops and webinars that we're holding. So if you have beekeeping questions or you want to get more involved, we hope you join a local beekeeping club, but you're also welcome to come join us at some of our workshops. Our upcoming office hour webinars are on June 17th, July 22nd, August 26th, and September 23rd. We have our YouTube channel where we post recordings from our office hours webinars. So if you want to rewatch what you're watching tonight, we'll try to cut out the technical difficulties and you'll be able to watch it there. And then as always, you can sign up for our newsletter by going to the website, hitting Stay Connected, and then Pollinators and Pollination. And those news digests go out about every other week. And if you are in Michigan and you have a question for us, you can use the Ask Extension form on our website. It also allows you to add photos, so we'll get back to you if you ask a beekeeping question. And as we mentioned earlier, so we have guest presenters. Today is Katie Brandt talking about sustainable agriculture, research, and education, which is abbreviated as SARE.
So hello, everyone. I'm Katie Brandt, and I have the great good honor of working with SARE, Sustainable Agriculture Research and Education here in Michigan. We do grants, and you can see that map on the right is showing areas, farms, as well as other research and education sites where people did some of the work related to SARE grants. Other things I do, I work with the MSU Organic Farmer Training Program, which happens here in East Lansing at the MSU Student Organic Farm, but also in Detroit at Keep Growing Detroit. And some of it's online, and we do field trips too, if anyone's interested in that. I know organics is not a great fit for beekeeping. It's hard to go certified organic with bees, but most people who do the program don't choose to certify as organic. They're just using a lot of the practices. I'm also part of the Michigan Transition to Organic Partnership Program, if anyone's looking for a mentor to go organic. So the grant that I wanted to dive into first is the Farmer Rancher Grant Program, and check it out. This is just the standard slide set for the Farmer Rancher Grant. Happens to have a beekeeper. And so these fund a farmer or two or more farms. If it's one farmer doing the research, it's up to $15,000. And if it's two or more farmers from separate farms, then it can be funded up to $30,000. And that is to do on-farm research and on-farm education related to that research. And they fund about 40 projects a year. That's here in the North Central SARE region, so that's in the Midwest, not nationally. Typically, they fund a third to a half of the proposals. So you've got to be competitive. It's not everyone who writes a grant gets them, but it's a better rate than many grants. The other thing is these grants are pretty easy to get. In terms of You don't have to know like grant language as much. You just have to make sure it really focuses on sustainable ag. On this first slide, I should have mentioned Sarah Franzak is the state SARE coordinator, and either Sarah or I are willing to help with brainstorming and editing, well, not really editing, reviewing and giving advice on your on SARE grants. So we can forward to the next slide. I talked a bit about the Farmer Rancher Grant Program. So again, we can go forward. So I know there's a lot on this slide and it's very busy, but just looking at the section of that's titled NCR SARE Grants for Farmers. So the Farmer Rancher Grant I talked about. Youth educator grants are also often a good fit for farmers and also for beekeepers like yourself. So these are smaller, they're up to $6,000 to teach youth about sustainable farming and careers. Both of these are due kind of at the end of the year, December for Farmer Rancher, November for youth educator. And again, you want to focus on the 3 pillars of sustainability for these. So financial, social, whether that's for you, the farmer, or the broader community, and environmental. And then the Michigan SARE Travel Grant, we actually have promised these all out for the year. There was a lot of interest, but these fund up to $500 to travel to a conference, field day, or training for farmers or educators. I won't go into the other SARE grants below, but if any of those look like they're a good fit for you, don't hesitate to reach out. Last thing I'll mention from this slide is SARE.org resources. There's books and videos and podcasts and many other things that you can find at that link. You can go forward.
Great.
So there's some of the books and bulletins in that image there from the SARE resources. Really, this slide, I was just thinking if anyone has questions about SARE, you can go ahead and ask them in the chat, and I would gladly get an answer to you. You're also welcome to email myself or Sarah at the email addresses listed there.
Thanks, Katie. I'll keep an eye on the Q&A in case anyone has a question come in for you. I was curious because you said that the conference travel grants, you can apply year round, but they're gone for the year. Do you know when they'll be available again, or what is the time frame on applying and receiving those grants?
Yeah, it's an annual year. So I would say apply anytime, but the funding might not come through until 2025.
Okay. All right. Thank you. Awesome. Well, we know a lot of beekeepers who are interested in SARE and excited about learning about the program. So thanks a lot for being here. I know you had a long day teaching new farmers, so we'll let you go and we'll pass along any questions we get to later for you to get back to later this week.
Thanks so much, Anna.
Thank you, Katie. All right, so next I'm going to talk a little bit about swarms. In case you're not aware, there are a lot of swarms out there, and I can tell you that because sometimes people call Michigan State University when they see a swarm and they want to know what to do about it. So just a couple of pictures of swarms. You know, swarming is the way that honeybee colonies naturally reproduce by splitting themselves. They can issue a swarm and then establish a colony somewhere else. And we as beekeepers work really hard to deter swarming by splitting colonies and giving the bees lots of space so they're not crowded. But it can be a really strong urge for colonies to swarm. So a couple of photos here. The thing that I really want to share with all of you is that as beekeepers, you are a resource for people when there's a swarm. So if you're not interested in catching a swarm or available to catch it, it's great if you can refer people to other beekeepers who can help. We want to try to catch swarms before they establish somewhere else because sometimes they move into a structure and can become a nuisance. So we have an article that we wrote for the public called "What Should I Do If I Find a Swarm of Bees?" Sometimes when people say they have bees, you have no idea if they're even talking about honeybees or some other insect, if it's a swarm or an established colony. So this article helps walk people through whether or not they have a swarm. And then it directs people to the Michigan Beekeepers Association's list for their swarm list. So if you're interested in collecting swarms, you can become a member of Michigan Beekeepers Association and sign up for the swarm list. And so we, as Michigan State University, we are referring all of these calls about swarms to the swarm list. All right, Megan, I'm going to turn it over to you for queen events and timing.
All right, so along with getting lots of calls about swarming, this is definitely the time that we get calls about queen events as well. So generally, the bees are going to replace the queen every year, or every year-ish, unless you do. So this is something that is not necessarily a surprise. It should kind of be expected. So it is best management practice to go and put young queens in every year, but we know that a lot of people don't or don't want to do that. So the message there is just to make sure that you are aware that it's going to happen. And, whether it's every year or every 2 years will depend on the health of the queen and then the amount of sperm that she has left. So that's really a factor of how many eggs she's laid and how well she's mated. So if you've had her in a little tiny nuke and she doesn't have, a lot of eggs to lay because she doesn't have a lot of space, she may live a lot longer. If she went through a really big colony, if she was in charge of a big colony through a big spring buildup, then, maybe 1 year is expected of her because she's laid all of her eggs. And so again, this time of year, so a lot of times if a small colony doesn't swarm, then that old queen gets replaced. Or if it's the swarm and you caught an old queen, a lot of times once she gets started, it's this midsummer, right after swarm season, that we see this queen replacement. And the reason that I'm saying expected happens is I get about 1 billion calls at this time of year of people that are panicking because their queen is gone. But unless you're replacing them, this is when you expect them to supersede her. All right, so we know that it's okay when it's queen right. We say that a colony is queen right when you see queen laid eggs. And we can tell that these are laid by a queen because they're right in the middle of the cell and there's one per cell. So that's the good situation. We would say that it's queenless if you see open larval cells. So they're only going to be doing this in a situation where they're superseding or emergency, or if they're swarming, which is separate. So you can't really say that the queen is or the colony is queenless unless you see this. So a lot of times I get calls and people say they need a new queen because they don't see the open cell, or sorry, they don't see the eggs. But sometimes the reason they don't see the eggs is because 1, they can't see eggs, or 2, they're nectar bound or they're pollen bound, meaning that the places where they would want to lay eggs are filled in with the incoming food, which is really common in a lot of places in Michigan, because we can have some pretty dramatic nectar flows. So if you think you're queenless, these are the 2 situations. If you saw open queen cells, or here we have a situation where a queen has recently emerged, a virgin has emerged, and there's one that's about to emerge. So these would be situations where I would agree that that colony is in the process of requeening itself and the old mated queen is probably gone. All right, so this is the situation that I was talking about with backfilling. So here you can see a frame that has lots of bees that are just about to emerge. So there's some capped brood along the edge. And in a good situation, if the beekeeper is staying on top of giving the bees space, then what would happen is that the bees would put all of the nectar and all of that pollen, on the edge of the brood nest, and the nectar would be above the brood nest. In this scenario, the beekeeper maybe didn't keep up or didn't have drawn comb, and the bees backfilled the brood nest with food. And what we want is that sphere of where the brood is to be always for brood. Except when we're preparing for winter. So at this time of year, as a new bee emerges, ideally the queen would go back and lay an egg in it. If we're not giving the bees space, or they don't have enough space, then the workers are going to come in and fill that all with food, and the queen won't have space to lay. And that's what triggers swarming. So you can get a crowded swarm even after this period of reproductive swarm. But a lot of times too, people will think that the queen's not in here in this context, but it's because they can't see the eggs because she just has nowhere absolutely to lay. So this is a situation which is, you both need to figure out your queen scenario and give them more space. You can go to the next one. So then the next one is showing pictures of torn down queen cells. Oh, sorry, go back to the, and so this is a situation that's really hard to determine because they could be torn down because they sorted everything out, or they are more likely torn down because one queen emerged and tore down the rest of them. So if you see something like this, it's likely that a queen event has happened. So on my website, I wrote this article. I should switch it over to an MSU document at some point, but I wrote it because I was selling queens and I was getting a million calls in the summer of people saying, help, I need a queen. And I just had one yesterday and it was exactly this scenario. And what was happening is, and this was before I was working at MSU, and I had people calling me saying they needed a queen. And I was like, well, what the heck is going on? So instead of just selling them queens, I said, I'll come to your house and install it. And in 100% of those situations in which someone called me thinking they needed a queen, they did not actually need a queen. And so what was happening is two things. One is that a lot of times the queen was just being in the process of being replaced, in which was a situation in which they just had to wait a lot longer than they thought they did and give them room to move that nectar out of the brood nest. Or sometimes it was a laying worker scenario in which a queen wouldn't be the appropriate. So this article is in there. The main thrust of the article is that it's a trap. You're going to say, help, I need a queen. And then you click on it and it's going to tell you why you don't actually need a queen in midsummer. And what you should be doing is waiting for them to finish requeening themselves. And the biggest mistake people make is not waiting long enough. So this figure really shows how long it takes. So they're going to start, so this is for grafting, but they're going to start, if you killed the queen today, they're going to start raising a queen cell out of like a 3 to 4 day old larva, 4 days after being laid or 5 days after being laid. Then the cell gets capped, and then the virgin queen emerges after 16 days after it's laid. So basically, almost 2 weeks, 10 days to 2 weeks after that queen is killed. Then she has about 6 days before she's ready to go on a mating flight. Then she has up to 2 weeks to go on a mating flight, depending on the weather. Then it takes her a couple days to get the pipes flow in the right direction for her to lay eggs. So we're talking about a month of queen replacement. And in almost all cases, most people were too early in looking. And what I highly recommend is that you put the date that you expect to see them, which is really, I would say, a month after you think that the queen was gone. And you don't dig in the hive until then. Because if there's a virgin in there and you go digging in the hive, or if there's a bunch of queen cells in there and you go digging in the hive too early, there's just a chance that you're going to mess it up. And it's very hard. Like, it is very hard to be patient. And I've even had people where I would say, right on the top of your box, I'll use Anna's example, like, Anna, do not dig in here until June 6th. And then she's not supposed to go in there until you would expect to see eggs or larvae. Because either the colony is requeening fine, in which case, yay, or what will likely happen is that there's a chance that the queen won't come back from a mating flight, but you digging in there is not going to help it at all. So it's either fine or it's not, and you just kind of have to wait to see how it sorts itself out, but there's nothing you can do by digging in there too early. So it is an exercise in patience, and then you can give them an extra super if you want to pull out the nectar from the brood nest, so when that queen does come back, she has a nice spot to lay. All right, so if you do not see eggs or a queen, assume the bees are replacing the queen and that it will all be fine. If it's not fine, the worst case is a laying worker scenario, in which case you'll do a newspaper combine. So if it's not fine, it can't get more not fine, basically. They're either going to sort it out or they won't. But you digging in there is not going to help them out with that. So this is the worst case scenario, is that they are hopelessly queenless. And this is a scenario in which the queen went out, or the virgin emerged, tried to go get mated, got eaten by a dragonfly or hit by a car, and came back, and then they didn't have any eggs or larvae to make a new queen. So that is what is called a hopelessly queenless, in that so they don't have a queen, but they also don't have the goods to make a queen. And then what will happen in that context is that you won't have any young brood to make a new one, but the workers can start laying eggs, which would get you to what's called a laying worker colony, which refers to the queen laying, or which is where the workers will lay only drone eggs. All right, which is I was gonna say, I knew Zachary was talking about laying workers, so I was trying to hold back from it.
Great. So Zach, you want to go ahead with laying workers?
Yeah. So that first slide was actually should be here. So Megan already went through the first slide. What is called hopelessly queenless colony. So workers have about 10 to 15 days they will develop the ovary when the queen's gone and when the brood is gone. Actually, even the capped brood, but mostly open larva. Open brood has ability to suppress queen raising, actually a stronger suppression ability compared to the queen. So in a normal colony, the reason the workers refrain from laying eggs is the combination of two factors: the presence of the queen, which has the queen mandibular pheromone that inhibits worker ovary development, and the second is the presence of a lot of open brood, and that's brood pheromone. Brood pheromone also suppresses worker ovary development. But when those two are absent, then the workers will develop the ovaries. The colony will be hopelessly queenless. So how do you tell if you have a laying worker colony? As of course, there's no queen in the colony, and then you see, compared to the slide Megan showed, there was one egg per cell. This one you see 5 or 6 eggs per cell. None of them dried up, so there are different stages. Some of them are falling down. So the workers just keep piling eggs into the cell, and this doesn't really happen with queens. Occasionally, when the queen run out of place to lay eggs, she sometimes will put 2 or 3 eggs per cell, but that's very rare. So this will be a good sign that it's a laying worker colony. And then of course, when eventually those 3 or 4 eggs per cell, the workers are going to weed out and leaving only 1 larva per cell, and those 1 larva per cell would be drawn because workers cannot mate and there's no spermatheca to store sperm. So all those worker-laid eggs develop into males, and I think my next slide shows mixed brood, but mostly I don't know why I probably forgot to put that. So I was supposed to put a frame of capped brood showing you the worker cells. Those are worker cells, but they are capped as drones because they cap taller. They are raised above. The regular worker brood would be flush against the normal cell depths, whereas the drone cells are just a tiny bit up, probably 2 or 3 millimeters taller. So if you dissect the laying worker bee, of course, it's very hard to actually observe a worker bee back into a cell. That's very hard to see. But I think 1 year I saw a colony has become laying workers. I grabbed a few and let's dissect some. So I saw this. So this is developed ovaries from a laying worker bee. Has about 6 ovules on each side, compared to 200 per side for a queen. The average is about 400 for the queen. So just to imagine that. I think I didn't show that. And then another very rare cases, I saw this only once in my life. is when everything is right, usually the laying workers have developed ovaries, but they don't quite smell the same as the queen. So maybe 1 out of 100 or 1 out of 500 laying workers will actually be able to produce that queen pheromone-like chemicals. So the workers are treating her just like a queen. So here you see as a retina, and this queen is actually packing herself into a cell laying eggs. That's 1, 2, 3. That's like nine workers surrounding that worker. So that worker is a laying worker, but she also happened to smell more like a real queen, so it's called a false queen. So if you take that bee and into the lab and run some gas chromatography, you'll see that worker has a lot of queen pheromone-like chemicals instead of smell like workers. So what do you do when you have laying worker colony? There's not much you could do. I would recommend beekeepers not to try to requeen it, because you pay $35 for a queen, and there's a 95% chance that your queen is going to be killed by those bees. All those workers that are laying eggs think they're the queen, and they're going to be very aggressive toward the queen that's caged. Most people just combine with another colony. If there was a newspaper, or more often, they just shake all the bees in front of a strong colony and remove the old box away. So then the bees have no place to go but stay into either the colony or drift to the next two colonies close to the original position of the laying worker colony. So you basically absorb the cost instead of trying to rescue that colony by investing in more queens. Occasionally, you give them a frame of brood. Occasionally, they could successfully raise a queen. That's from a queen cell. Now you can wait like a month, as Megan mentioned. So it's most of the time, by the time you found the colony has been laying, it's already 2 weeks, maybe 2 frames of bees. So it's not really worth the trouble, I think, to try to get a queen. Why a colony will take 5 or 6 frames. So, okay, I think I'm done with my section.
Great. Next, Dan is going to talk about drawing comb and managing space.
Yeah, so when I think about, bee season and it's swarm season, I also think about this period we're in right now as wax making season. This is the prime time to get frames drawn out, and it's really important to utilize this window because it's not open forever. So our bees make wax. It's the young bees that are able to make the wax. They have glands on their abdomens, and they secrete wax. When they secrete it, it's brand new, sparkling white, and they're molding it into the combs. But it's only a narrow age class of the bees that are making the wax. They're in I'm seeing some slightly different numbers published, but they're roughly 12 to 20 days old. So it's not until they're into that end of that second week, and it really only lasts about a week, kind of that third week of their life is when they're really at their wax making peak. So if you think about your colony demographics, it's that age class that's going to make the wax. And to do that, they're essentially digesting honey. It does take a lot of calories for them to create wax. So you need really well fed bees. Now, fortunately, right now, I'm going to say we're kind of in the midst of this wax drawing window, and where I'm at in Ingham and Clinton County, we had a really good dandelion flow that kind of closed out April into May, and we're in the midst of a really good autumn olive nectar flow right now. So the bees are drawing comb really well without any supplemental feeding. But some years, if weather is marginal, if there's not good flying weather, there's not good nectar flow, supplemental feeding of syrup can help your bees to draw comb a little quicker. If, again, the environment provides it or you provide it, but they need to be getting that sugar somewhere to help them make wax. So hopefully what we see when we put these new frames of foundation in, we want to see these nice, beautiful, every cell is just perfect geometrically. We don't have any little deviations or pieces of burr comb or things that are they're not really problems for the bees, but us as beekeepers, we kind of like things neat and orderly. So when you see, again, we've got this really nice young blonde wax here that's brand new. It really makes me happy to see that in the spring. It's a sign of good health. It's a sign of colony growth, and just like, pay attention to the landscape and the flowers, certain things getting greener in the landscape tell you it's spring. When bees are making wax, that's really a cue to me that like we're in kind of peak springtime conditions. So I'd really like to see this. This also, you do get, again, bees are making wax. This is not a problem for the bees, but me as a beekeeper, this annoys me because they've put all this this effort into producing this wax and sculpting it, and they haven't adhered to the foundation. They made these kind of lobes of burr comb and cross comb, and sometimes it's a case of bees being bees. But one thing you can really do to minimize that, if you've got a lot of comb to get drawn and you have a lot of frames of foundation in the colony, is making sure you get them really snugged up tight together so that bee space is appropriate. If you've got them spaced too far apart, you quite often will get a situation like this where the bees, if there's a large volume, they're going to fill it with comb. They're often going to fill it with drone comb, burr comb. Here it looks like it's a mix of drone and worker cells, but they're just filling it with nectar. So having things tightly spaced while they're initially drawing it and getting established nice clean sheets of comb is important. Also, if you're using foundation like a plastic foundation that's pretty common and it is not well waxed, then the bees are not really treating that as much as like a starter template. They're just using it as a structural anchor, but they're not as likely to build out that nice hexagonal pattern like we like. So if you don't have well waxed foundation, you can apply an additional layer of wax yourself, and that'll help promote the bees building where and how we want as beekeepers. The other thing when it comes to drawing wax in this time of year, kind of making wax and managing space, is Megan talked about this a little as it relates to swarming. It's an issue in this regard as well, because when we see this backfilling in the brood nest in particular, again, that's nectar coming into the brood nest where it's crowding the brood nest, colonies getting congested. One, it can push the colony towards swarming, but one way you can sort of mitigate this as a beekeeper is giving them more frames. And if you don't have comb, drawn comb, even giving them frames of foundation, particularly this time of year, is, like I said, this is a really good window for bees to make wax. So if you're getting foundation kind of adjacent to the brood nest, bees get into that wax drawing mode, and it gives them, one, they're consuming some of this nectar to have the calories to produce the wax, but they're also, in making that wax, they're expanding kind of the skeleton of their colony, so they have more appropriate places to put that nectar where it's not going to clog up the brood nest like that. So when you see this backfilling, good indication you want to be adding more space, whether that's new frames of foundation, supers. If you've got drawn comb, great, but a lot of times that's not a luxury. We have a big inventory of drawn comb up our sleeve. So another thing when you're thinking about looking at managing space in the colony and when to give them more space, particularly in the spring, being a little early is much better than being too late. Again, you're going to induce that crowding, that potentially swarming, not from a reproductive swarm, but a crowding swarm. And when you see comb like this between boxes, that's an indication they've been on a great nectar flow, but they probably should have had another box or two, weeks prior to this to give. It's not going to totally prevent this, but it is going to mitigate it by giving them that space to put it, again, typically above the brood nest. That's where they want to put the honey. When you see this, one, it's really challenging to work this hive and not squish bees and end up with a huge sticky mess when you've got this amount of comb between the boxes. So giving them space, colonies are in growth mode this time of year, trying to be out in front of the bees as much as you can when they're in the spring conditions. And then we just, this is another resource article by Megan about really the value of drawn comb. It is really incredibly valuable to you as a beekeeper. What you can do with your colonies, the options you have for managing them this spring, giving them that ready-made place, whether it's to store honey or the queen, if you're making splits and you have somewhere the queen can lay into immediately, that's really valuable. So protecting that comb, storing it in the off season, is something you really should, I guess, value and protect, because it's a significant investment for the bees to make it. So it is valuable. You should look after it. So you read more on why that is here.
Great. Now we're going to turn it over to Megan for European foulbrood.
So after all of that like happy talk about springtime and summer and growth, I'm going to talk about all the things that can go wrong with diseases. So this is a resource that we have that I'm incredibly proud of. It was done by a whole group of people at a few different universities. It's diagnostic tools for honeybee medicine. It can help you diagnose, and it tells you all the way to field diagnose different diseases. It has lots of photos. It has fun games on disease diagnoses. It's available at Bees Need Vets. You can also get to it from our website, the pollinators.msu.edu. So when we're diagnosing European foulbrood, there's a couple things that you can do. The reason I'm talking about European foulbrood right now is because it does have a very, very strong seasonality, and this is the time of year for it in most parts of Michigan or northern states, really, is this late spring. And so the way that we diagnose it, one of it is just being, is it showing up in the late spring? And so that makes it most likely European foulbrood. The two things that you can easily mix it up with is American foulbrood or varroa, the parasitic mite syndrome. So if it was American foulbrood, you'd have a positive matchstick test, meaning that if you put a matchstick in, you could draw it out over 2 centimeters. And then there's another test called the Holt's milk test, which is super easy and cheap that you can just do at home. And those tests are very specific for American foulbrood. The other thing is that American foulbrood looks pretty different. And so that's what that website is really good for, is showing you those visual differences. The parasitic mite syndrome, which is caused by the viruses transmitted by the varroa mite, can look pretty similar in that the larva, these older larvae, will kind of get melty. If we want to be fancy, we'd say it's a loss of internal pressure because they just kind of disintegrate. With the parasitic mite syndrome, though, you can rule that out because you'd have a high mite load. So if you did an alcohol wash, you'd be able to see that, or if you haven't been managing. And that's usually in Michigan, we generally don't see that until August or September. So if you see some weird brood at this time of year, it's likely European foulbrood. If you want to confirm your diagnosis, though, you can use that website to do a visual diagnosis. And that is, I can look at a colony and say this has European foulbrood based on the visual signs. It is pretty specific. You can also send it to Beltsville. And if you just Google Beltsville lab, Beltsville honeybee lab, it will give you directions on how to send a sample in. But that is a free national laboratory. They can actually test it. They can give you a confirmation. There's also an ELISA or a commercial test. There has been some availability issues, but that may be still available, or you may be able to find that, which is basically like a COVID test. So you can go to the visual. So the way that we diagnose it visually, one of it is it's highly variable. So American foulbrood is, the one that a lot of people are more concerned about, but that one is kind of more consistent. The larvae always die in a particular way. I love this photo because it shows all of the variability. So right in the middle of the screen, you can see some that are misshapen and they're going in a corkscrew. You can see one that looks pretty good, but it has a little yellow of the brood food. You can see one that's kind of in the center, but down a little lower, where it looks like you can see the skeleton of it, and that's actually the trachea. And you can see so anything that gets yellow or brown, and you see corkscrew shapes, and you see meltiness and all these different ages, that's generally going to be European foulbrood when it's this diverse. And the reason it's so diverse is because it can cause the larva to die at different ages. And also we can get a lot of different secondary bacteria involved. So it can look very, very different. So if you see a disease in your colony, or you see brood that is yellow and brown and twisted and melted and chewed down, and lots of different things, that's likely going to be this bacterial disease, European foulbrood. So these are the categories that we give it. Again, here's another lovely photo from Rob Snyder that shows just how diverse it can be. So you can see this twisted or corkscrew shape. That's pretty characteristic to American foulbrood. You can see some yellowing. It can dry down to a scale. The difference between the European foulbrood scale and an American foulbrood scale is you can actually remove the European foulbrood scale. You can have holes in the capping. So in this photo, right in the middle, we can see a sunken capping. A lot of the stuff on the internet, or yeah, perfect, or in online will say that American foulbrood is capped brood and European foulbrood is open brood. In reality, they both can be both. So you'll see American foulbrood is both capped and open, and European foulbrood is both capped and open. We talked about them being just straight up melted looking. And then again, I mentioned the visible trachea, and there's a really good example in the bottom center of this where it looks like you can almost see the skeleton through. And that is very characteristic of European foulbrood. All right, so there is not an exact treatment that works for everybody because we have lots of different situations. So what treatment you should do really depends on your situation. And this is something that I'm happy to answer in the chat. And then also you can answer through our questions thing. I can help people deal with their situation if they have European foulbrood. A big part of it is how many colonies you have. If you're dealing it with 2 colonies versus 2,000, those are very different scenarios. But the big thing is the chance of reinfection. So you don't want to take a drastic measure if your chance of getting reinfected next year is really high. And what we talk about high chance of reinfection is how many beekeepers are near you. The USDA just published a paper that matches with our own data that shows about 30% of the colonies that they're investigating has the bacteria that causes European foulbrood. That's very, 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 very high. So, you don't want to burn all of your equipment and then next year you get a colony and it's going to get the bacteria again, and then you're going to burn all of your equipment. However, if you lived in a really remote location and you have a closed apiary, maybe burning all of your equipment would actually make the most sense. For a lot of people, you can do watchful waiting, and a lot of times the colonies just pull out of it. Or you can use antibiotics and working with a veterinarian. And I know there's even veterinarians on this call tonight too. So we can talk about that as well. So it does really depend on what your specific situation is. We do disease management at the yard or operation level. And that's because for every colony that you see with active signs, we have lots of studies that show that there's going to be the bacteria present in all or most of the colonies in that yard. So with some diseases like American foulbrood, you can say, this is my one colony that's infected, the colony that I see the signs in. But with European foulbrood, if you see this one colony with the signs, you can assume that the bacteria is going to be most of the colonies in the yard. So if you only quarantine this one or treat this one, you're not really managing the spread of the disease in their yard. The other thing is we're working on some studies that show that the pathogen, Melissococcus plutonius, is very persistent on your equipment. So if you have it in your operation, it's kind of in your operation at that point. And so then we switch from trying to eradicate it to just making sure that we're controlling the disease and that it doesn't get out of control. All right, and last October, Peter Fowler, who is a veterinarian who is completing his PhD right now, and I hosted a webinar. I think it's like an hour and a half. Oh yeah, you can see it's an hour and a half long, all EFB. So if you're like me and you just can't get enough of EFB, this is basically what he and I have been working on the last 3 years. And we've presented some of our projects on there and what we know about EFB. And so we can do a deep dive into it.
Great. All right. And then another thing to be thinking about, as always, is varroa. So, we have lots of resources. We try to compile them from MSU and then other places, universities that are trusted resources on varroa mites. But if you're new to thinking about varroa, you want to start thinking about it now. You want to start monitoring your colonies so you get an understanding of what that looks like and you get some hands-on practice monitoring, and you want to make a plan for managing them throughout the season. Our resource that's really nice is from the Honeybee Health Coalition. It's their Tools for Varroa Management Guide, and it gives a lot of background on varroa and also goes through all of the different management options that you have. Normally we're trying to monitor colonies starting in May. It's good to start early in the season to practice. Sometimes it's not as easy as you think it's going to be to collect bees, a sample of bees, and do a test on them. Most of the time we're doing an alcohol wash. The powdered sugar roll test is another option for you if you aren't comfortable with the alcohol wash. And there's lots of videos online. If you're not sure what I'm talking about when I say an alcohol wash, send me a question through Ask Extension and I'll send you some videos so you can see what that looks like. But the main thing is we're trying to collect about 300 bees in our sample, and we are, with the alcohol wash, putting isopropyl alcohol in a jar with those bees. We're shaking them, and then we are pouring the alcohol through a sieve so the bees don't get through, but the mites and the alcohol get through, and we can count how many how many mites are in our sample. We also really encourage you to count how many bees are in your sample, because we know that a lot of times beekeepers have a tendency to undersample. So it'll give you more complete information to count the bees too. So again, lots of videos there. And then we're thinking about our treatment options. So we know that we can just expect mite levels to continue growing throughout the season. We want to keep mite levels really low now to try to prevent some of that varroa mite infestation growth. So if you have colonies with honey supers on, which many of our colonies are collecting honey for human consumption, there's a few options. One is formic acid, so that's Formic Pro or Mite Away Quick Strips. And those are going to be really good options if you have a lot of sealed brood in your colonies and you have honey supers on. There are some restrictions, or especially around temperature. So with all of these treatments, you will want to read the labels carefully and figure out if they're a good fit. But formic acid is normally going to be our go-to in the summer with large colonies with sealed brood and honey supers on. HopGuard's another option that you can use, and that's also allowed with honey supers on. And then the other one, oxalic acid, is mostly effective when you don't have sealed brood in the colony. So there are some cases, especially with Megan's talk about, the queenlessness and trying to figure out when you can expect a new queen. Sometimes what we'll do is look at our calendar and figure out when we expect to have a new queen who's laying, but before there'd be sealed brood in the colony. And that would be an option to use oxalic acid then. If you have a small colony, so if you just, purchased a nuke or installed a package colony, then you're going to want to look at some of the treatments that are appropriate for smaller size colonies. So those are going to be HopGuard, oxalic acid. Another one that you can use is Apivar. Some beekeepers prefer not to use that one because there is some new data now showing some resistance to Apivar as a treatment, but these are treatments that you can use with smaller sized colonies. So again, all of them have kind of their right place where they're more effective, less effective. Some of them have temperature restrictions. You really want to make sure you're very familiar with your options and you read the labels, but these would be some options for you. So, if you've got a nuke, you could consider doing a HopGuard treatment. If you got a package, you could do an oxalic acid treatment after the new queen has started laying, but before there's sealed brood. And those are a couple of options for you. All right, so there should be a program evaluation survey that comes up when you close tonight's webinar and in the follow-up email. So feel free to share any comments there. And again, the resources from tonight's webinars are on this webpage, the May video webpage. So there's the QR code and you'll find the links there. And with that, we are going to jump into questions.
All right, so we just have a few questions on here, but there's some good ones right now. So, Anna, I'm going to go right back to you on a question we do get a lot. With the alcohol wash and the sugar roll, the question is, don't most of the bees end up dying in the sugar roll anyway?
I mean, so that's the question is the alcohol wash, the bees definitely die. The powdered sugar roll test some beekeepers prefer to use because they don't want to watch their bees die in front of them. Immediately after a powdered sugar roll test, some of them are still alive. I do know that there was a graduate student tracking the lives of bees after a powdered sugar roll test. And I don't know if I've seen the paper published on that. So I don't know for sure. But if you can get comfortable with the alcohol wash test, it's a little bit more foolproof. There's some issues with the powdered sugar roll test. If it gets too hot or if there's too much nectar in the sample, then it gets too sticky and it might be a failed test. So in general, we're saying if you can do the alcohol wash, do it. If it's kind of too much for you, then just do a powdered sugar roll test. We just want you to be testing for mites.
All right, and then Dan, I like the way this one is worded. It says, if drawn comb is gold, what is dirty old comb worth? What to do with grungy old comb?
Yeah, that good point. And there is a diminishing returns as it gets older. It is good practice to cycle out that old comb. So I would say the best thing you could do with it was if you have a solar melter or something, and you want to render the wax out of it, great. But yeah, that's a fair point. The old combs accumulate a lot of things, both the bacteria and things like that from the environment. So cycling it out, I've seen different recommendations, but somewhere, if you're somewhere in the vicinity of like 10 to 20% per year, it's only a couple frames a box. That's not a lot, especially when you think about if you're splitting colonies, making new colonies. Again, you're probably replacing that with foundation. So, yep, it's good, to say, but when it gets past that, 5-6 years old, it starts getting real black and really dark like chocolate. Yeah, you start thinking about purging that. I typically will move those older frames like out to the perimeter of the colony. So when I do want to purge, I'm not purging like a frame of brood. I work them out to the outside, and then next stop is either the burn pile or the wax melter.
Zach, here's a question for you about the laying workers. So how viable are the drones that are produced from a laying worker hive? So if they mate with a queen, what would the impact on the queen quality and diversity will result?
There's actually a study. Interesting. When I was doing my PhD comprehensive, this question came up, so I had to think on my feet and design an experiment to test whether drones produced this way can participate in mating. So there was actually a paper maybe 4 or 5 years ago showing that those worker-produced drones can participate in mating. In terms of consequences, there's really no bad consequences. It's just the workers are just having the genes from her parents passed on down to those, so there's no bad consequences. It's not going to transmit a laying worker gene. So if you're trying to look for that.
Yeah, it's almost like a happy ending. Like they died out, but some of their genes lived on through that plucky, tiny little drone. So someone asks, if you want to keep your queen for a second year, what traits or aspects of the queen would you consider in your selection? Age, brood pattern, hive behavior, etc. So I'll answer first, and then I'll let other people chime in. So sometimes I get very fancy queens because I'm a sucker for very fancy queen genetics, and I like to try new ones. If I have one that's expensive, like an II queen or something from a breeding program, and I want to keep her for a long time, the thing that I think makes the biggest difference besides a good healthy hive is to keep her in a small hive. So if she runs out of sperm, and then also if you think about her egg laying is an enormous metabolic cost. It is a boatload of protein and fat that she has to pull out of her body. So if you can reduce the number of eggs that she lays, so for the ones that I want for queen rearing, I want her to lay, enough eggs for me to graft from, but I need 90 a day, compared to the 1,500 she could potentially lay a day. So what I tend to do is keep them in nukes or, 3 tall nukes so that she doesn't have this huge brood nest where she's laying thousands of eggs a day. The thing that often makes them die is because they run out of sperm. She's going to go out and mate. That's all the sperm that she has for her life. And then, that's kind of it. So, so for me, if I have a queen that I want to keep for genetic reasons, I'll put her in a nuke and restrict the amount of egg laying she can do. All right. So Zach,
oh.
go ahead, Anna. Yep, if you have an add to that, go ahead.
Oh, that's okay. So I mean, I think it was just beekeeper preference in terms of what kind of genetics you want to hang on to. But I'm really more and more these days appreciating the gentle temperament of bees. So that's definitely something to consider. And then just overall health and how the colony builds up and looks, we consider those things too. And sometimes beekeepers want to hang on to the old queens and they'll put them in a small nuke and requeen the colony with a new younger queen.
Yes, that's a very good point. And I just worked with some queen producers down in Texas last week, and we had to pinch all their old queens. And I told them, I was like, it's a good thing I didn't have a pocket full of cages. I would have flown home with pockets full of queens. It was hard to do emotionally. I am a softie and don't like pinching queens. But I also understand that the best chance for a colony is to have a young queen. So you could take a queen out, put her into retirement in a nuke, let your big colony requeen itself, and that's, I think, the best for everybody. Zachary, if a colony is hopelessly queenless, would they be bringing in pollen?
Says a very limited amount. There would be some foraging. There's actually a sort of division of labor still in the colony. But the workers are laying eggs, usually do not forage that much. So they are into reproduction. But there are other workers, there's some kind of social feedback. So then once there's enough laying workers in the colony, there will be maybe, I would say 10% bees still keep foraging and not really becoming laying workers. So very little compared to regular colony. They're already pretty weak. So they could be bringing some pollen, some nectar to just keep that colony barely alive. And yeah, they don't store a lot of extra nectar. They're not going to make a capped frame of honey, for example. They just have a little bit of honey on the corners and some pollen, just barely enough to keep that drone brood going. So you will see a few pollen forages.
Yeah, it's definitely much as I have heard people say, oh, you can see pollen going in. That's how you know it's queen, right? And that's a myth. You can definitely have a pollen going in, but yeah, Zachary's, it's just going to be a weaker colony overall. So I'm going to go to Dan again, and it says you talked about adding wax to plastic foundation. How does one go about doing this?
Yeah, so I haven't done a ton of it. I usually buy it, and from bee supply companies, you can pay for double waxed or triple waxed. I find that to be worth the money, but I have waxed some foundation myself. I will say, because you're heating beeswax, definitely it's an outdoor or a garage sort of job. Probably also want to have a fire extinguisher handy, just in case. What I've used to do it is an electric fry pan or like a crock pot. You want to hold it, melted, but you don't want it to be too hot. So you want a reasonable thermostat on that and keep it just above the melting point. As far as applying it, what I've had the best luck with is they're like the foam type paintbrushes. Like I've seen it called the speed brush, a little rectangle you can paint like in the corners and that that works pretty well. I would imagine a small roller would also work fairly well. But if you're putting it on like that, you're gonna get a thick coat of wax on. Generally, the cooler the wax is, like just past the melting point, you're gonna get a very thick coat. So you can experiment with it a little. You don't want the wax too hot, or you're actually going to like warp and twist the foundation. Or if you've got the one piece all plastic frames, I definitely have seen those like they'll flex and then they don't twist back. So getting too hot can be an issue. But basically, just melt it and paint it on.
Excellent. I'm gonna go back to Zachary. It says, if you shake the bees out, what happens to the nurse bees that have never been out of the hive? Will they know how to get into another hive?
Yes, that's actually a beekeeper's secret. As you shake a frame of young bees that have never flown in front of another hive, all those bees will just walk into that new hive because then assume they never had flight experience, did not have orientation flight yet. So those bees are actually 5 days and younger, about mostly. So then, yeah, so they'll just go to whichever colonies you should. It's actually the bees that have flight experience, like the few pollen foragers and act foragers will actually try to fly back to their old location, and then they will give up after maybe a day or two. But I have seen some bees very persistent, like 2 weeks after I moved the colony, foragers are still waiting, looking for a hive to enter, and usually they're very defensive, trying to say, so yeah, so you actually have to worry about the ones who's experienced that will refuse to go to another hive. But I would say for the late worker colony, it's not a big concern. It's already weak, pretty weak, and then they'll be happy to join another colony.
Yeah.
But what you do not want to do is to shake it in front of already 2 week colony that you could risk your queens being killed. So if you have a baby colony with 2 frames, you either you made a 2 week a split or winter colony. I've seen some winter colony just barely made it in 2 frames, and it would be a mistake if you shake in front of one of those. And there'll be 3 frames of laying worker bees compared to your 2 frames. So then no women in that new colony that could actually seek out the queen and kill her. So you actually lose something instead of getting the bees. So shaking in front of a bigger colony to absorb them.
Yeah. Dan, there was a follow-up to the coating of the wax frames, and someone says, will rubbing a wax bar on the foundation work as well?
I've heard beekeepers do that. I've not done it myself. So that I don't know. I don't have firsthand experience with that, but I've heard it is a thing. So quite possibly.
Same for me. I only use beeswax foundation. I don't use the plastic, but I've heard people will like melt a chunk of beeswax, like soften it a little and rub that on there. I assume it would work. The goal is to have some wax on there, but I don't have firsthand experience. I'm going to take this next one because this is my favorite topic. It says, I run 2 yards and wondering if I should add some diversity with a new queen from another beekeeper. This is a preference. So if you have really good bees in almost all places of Michigan, unless you're in a really isolated area, if your bees are going out and getting mated, there's very few chances of actual inbreeding anymore in the US. If you are in a really remote area, then yeah, it is useful to switch out or bring in some new genetics every now and then. But also it's very fun to bring in new genetics. And for me, one of the most interesting parts of beekeeping is working with bees that are really different. And so, I have my bees, which are obviously the best and I love, but then I also love to get, try and breed bees that are coming out of different breeding programs, or bees that have hygienic behavior, or bees that your friend really likes and that works really well, or it is really fun. So in terms of, is it necessary? It's probably not in most places unless you're incredibly isolated. But is it very fun and makes you a better beekeeper because you get to work with new genetics? Absolutely. So it's my recommendation that it is fun to try them. All right, Anna, have you treated your hives for mites this year? What method did you use, or when do you plan to treat next, and with what method? And Dan can chime in too after.
Sure. So not yet for the spring treatment, but it's on the list of upcoming things really soon. So what I'm planning is for the colonies that are large enough in size. So we have some colonies in double deeps with honey supers on. Those are going to get a formic acid treatment. I like doing the 2 strips at once just so I don't have to come back later and do the second strip later. That's just my preference. So that'll probably be a Formic Pro treatment. We have some small colonies, so we could consider like a hop guard treatment for those. And then there is a yard that we just requeened. So we're going to be like looking at the calendar and figuring out exactly when we expect the new queen to be laying before there's sealed brood. So that we can kind of maximize our mite treatment in those colonies. And that would be a formic acid or an oxalic acid treatment, but there wouldn't be any sealed brood in the hive.
Dan, did you want to add anything?
No, that's pretty, so we haven't treated this season, depending on when you count the season. The last treatment we put on was oxalic acid in the dribble, coming out of winter in, I'd have to look, but probably the last of February, give or take. It thought being before they start brood rearing this year, there was a little brood in the colonies in February because it was just a mild winter, but that was the intent. So that's the last time. We have split fairly heavily, so that does put a dent in the mite population. But yeah, as Anna said, we'll be getting into formic as the primary upcoming treatment here.
And I'm the same as that. I count kind of my splits. I also did some walkaway splits, which for me counts as part of my mite management strategy. I don't want to ask Zachary because he's our resident mad scientist who tries to get colonies with lots of mites for research. So I feel like he gives some, he maybe isn't the person, he's trying to grow mites for that. But I am waiting till everything gets requeened and bigger before I start anything. But I will say that I have already purchased my mite treatments for the year. All right. I got distracted because I can see Reno in the background of Dan's thing. So there is a question. What a handsome boy. So it says, I've seen some videos that using Dawn dish soap works better than alcohol. Have you tried it? I'll answer, and then I know Dan's had experience too. So I don't like using dish soap in the field because it's slower to kill the bees. So Dan and I are both on a committee that writes recommendations on how bees should be killed. And one of the things you consider is how quickly that happens. And the alcohol, if you use, the 70% is definitely much faster. The dish soap is quite slow. However, we use the dish soap quite a bit in the lab. So what we do is we actually take a scoop of bees into a plastic baggie. So the baggies, if you use the Ziplocs, they have kind of a stiff top. And we scoop the bees into a baggie, and then we put them on ice, and then we put them in the freezer so they're dead. And then we wash them with the dish soap back at the sink in the lab. And a lot of times that's because we're doing 100 hives per yard. So we have a lot more to process. So it makes sense for us to do it in a lab versus at home. But they're both perfectly fine for washing the mites off the bees. The alcohol tends to kill faster. Dan, you've done 1 billion mite washes in your years.
Yeah, the only thing I have to add is so when I do alcohol, I pour it out in like a tub and then look. I know some people look up in the jar and look from the bottom and count mites. That works The issue I have with the dish soap is it's foamy. So if you're pouring them down into a tub and then trying to look and see how many mites you've got in the bottom of that tub, and you've got a layer of dish soap froth on top, that's challenging. I've seen, some of a mirror. It seems like overthinking it to me. The alcohol works and it doesn't have the foaming issue. So that's my standard method.
Yeah, and when we're doing the dish soap back in the lab, we're doing it in the sink using a boatload of water into screens. So we're washing, so we kind of use the dish soap to get all the bees off, and then we wash them clean with a boatload of water, which is more water than you really would have in the field. But they both, and we use the Dawn Ultra. I know Randy Oliver has done a lot on this. And they both kind of work fine, but for the ease of viewing and the speed of killing, the alcohol is kind of better in the field. So there's a question, I'll open this up if anyone wants to do it. Can you speak to the management of chalk brood? Is there a treatment for this? Actually, Anna, do you want to start it?
Sure. So a couple things. One is, chalk brood can be common in the spring, and it often clears up, especially with like a strong nectar flow. So you can just keep an eye on it. If there's not that many cells, you can just see if the colony recovers. But one option you have is that bees that are selected to have hygienic behavior are going to be more resistant to chalk brood. So you could consider requeening that colony with a queen that's been, selected for hygienic behavior, and that could help deal with it in future seasons.
And it's a fungal disease, so there isn't anything, you can't put antibiotics in. We don't actually have bee antifungals, which is, you need more research on that. So I'm going to start with the next question. And it says, for propolis, what type of hive setup would you recommend? And this person, Sophie, if you want to follow up, I'm guessing, or I'm interpreting this, that you want more propolis, which may not be what you actually want. So previously, we used to find propolis just really annoying. And then Marla Spivak from Minnesota and Mike Simone Finstrom and Maggie Shanahan, there's quite a few people that have done some research looking at propolis and found that it's actually quite beneficial to the colony. And then we are actually starting a project with the USDA this year looking at boxes that have extra propolis because of this potential beneficial properties for managing disease. So if you're asking how to get more propolis because we like it, what the USDA and the Minnesota lab found is that if you chip kind of the inside of the boxes and make it really, really rough on the inside, you can promote the bees to having more propolis. And you kind of create what they call the propolis envelope, where you have the whole inside coated with a heavy layer of propolis, and that's thought to reduce disease. In terms of it, and the reason they went with that is because if it's on the walls, you'll have lots of propolis without it being between the boxes and super heavy and annoying in between there. So I don't have recommendations on how to minimize it because I am definitely in maximizing mode right now based on, the research that's come out showing how beneficial it is for disease management. Oh, and Anna just put it in the chat. Yes. And I love an article with a snappy title. So they call it Thinking Inside the Box. So we're actually using the exact same boxes that came out of that study to see how propolis affects virus levels and bacteria disease levels. All right. Dan, so this person says they just had a swarm. They tried their best with adding space. What is the best? I'm going to actually change this to a two-parter. So it says, what is the best next steps? So I think they mean for the colony that just swarmed. And then Anna, if you want to say what they could do next year instead of just adding space. So if Dan, if you want to do what they should do now, and Anna, what they should do next year.
So if the colony swarm, the best thing you can do, and this goes back to early on this evening with Megan, is kind of the best thing you can do is leave it alone for now. If it's issued a swarm, that means the existing queen is left with half the bees, give or take, and it's left behind a virgin queen that was, probably hatching out within a couple days of that swarm leaving. So you going in and messing with them, there's nothing good going to come of that. If you know when they swarmed and can kind of do that math, whether you actually saw the swarm leave or you did an inspection and realized, oh, we've got open swarm cells, and you can kind of read the brood nest to see, like, basically when is the youngest brood in that colony. You can do the math and say, okay, I know the swarm left on this date, and then count down your time for your virgin to get out and get flying and get back and mated, get back, start laying. So best case, a colony post swarm, leave it alone for a few weeks. Let that virgin get mated and settled in, and it's going to be, then you can go back in and see either, yep, success, new queen underway. If you wanted to, given we're kind of in a nectar flow with probably more favorable conditions coming, you could throw a super or something on top, just so when that new queen does get going, her brood nest isn't totally clogged up. It does give the incoming nectar somewhere to go in the meantime. But probably hands off for a couple of weeks is best practice.
All right, and then for next year. So coming out of winter, you're going to be checking your colonies. You're going to be trying to give them some more space. And by space, we mean empty drawn comb above the brood nest so that you can add your honey supers early. If you have extra brood comb that's, drawn from a dead out, or if you left your bees in a few boxes, you can rearrange, pull equipment out of the shed and try to give them empty drawn comb on top. And you're checking your colony, and you're ideally doing your split before you see signs of swarm cells. So we have a lot of past webinars where we look through some of the signs, backfilling in the brood nest, but before they start building those swarm cells, you're splitting the colony, and then you're giving them more space, which means empty drawn comb. We can go into too, like if you do start seeing swarm cells, it'll depend if the colony is left or not. And so then you'll have to kind of adjust, and I can go into that if you want, but ideally we're doing all the splitting before we see swarm cells.
Yeah, so in some places you can get away with just giving them space, and there is a genetic component to that, but in most places, like in Michigan, because our nectar flow comes on so strong, it's really, you almost just have to split. And even then, sometimes it's not sufficient. All right, so someone did point out the negative point to me being so overly enthusiastic about bringing in other bees. And they said, if you have healthy hives, does bringing other bees and queens increase the chance of diseases? And that is absolutely true. And like, my love for bringing in new genetics is almost equaled by my love of talking about biosecurity. And so if you do bring in nukes, or if you bring in packages, like, you absolutely are bringing in diseases. If you bring in queens, you can absolutely bring in diseases. I've even seen mites in queen cages. I've seen small hive beetles in queen cages. So it's a non-zero chance. If you're just bringing in mated queens, then it's much smaller than if you're bringing in nukes or packages every year. So I do have a closed apiary, and I try not to bring in stuff. And I'm very careful of where I would get my bees from, but there is absolutely a non-zero chance that you can bring things in. So if you're in an area where your queens get, well mated, well, if you're in an area where your queens get well mated, but then there's also a chance that they're going to get diseases from somewhere. So no, nobody's safe. We're all going to die, I guess, is the main message. So, but yes, there's definitely a chance for that. Anna, regarding the OA treatment for mice, can you comment on using OA vaporization instead of the drip method? Is that more effective, or is it gimmicky with new high-tech vaporizers?
Yeah, so I don't have much experience at all with oxalic acid vaporizers. The main reason is there's just added health risk for humans if you're using a vaporizer. So you do have to wear a respirator, and there is some concern that if your respirator isn't done correctly and you inhale those vapors, it can be damaging to your lungs. So that's why I opt for the oxalic acid dribble or solution method. I understand that the vaporizer can be very effective, but also the dribble or the solution is really effective as well. With the vaporizers, I've heard that there is a little bit more variability in those products. So that's another thing to consider.
All right. Actually, Zachary, I'm going to have you do this one. So I've never done this, but if you pinch a queen, have you ever put it in alcohol and used it as a swarm lure? Do you think that will work? And you're muted, Zachary.
I know some beekeepers try to put squash queens or lime queens into alcohol and then save that cocktail for something, like a queen pheromone strip. You put a few drops of that onto a piece of cotton and then put it inside a hive. It's if you have enough queens. I haven't heard beekeepers using that as a swarm lure. Swarm lure is more complicated. It has, I think, mostly nasal pheromone and maybe a dash of some queen might help, but it's not the major thing that workers are looking for when they're looking for a new nest. They're looking for the waxy smell, whether there's previously bees lived there and stuff like that. So queen pheromone could have a small role. But I know when Africanized bees were trying to come, USDA has spent millions of dollars for those tribes that have a lure inside. That's the main components, actually not queen pheromone, but nasal pheromone and a few other things.
Mm-hmm.
So I would say it's useful for other purposes. Like your nuke you make some nukes and the US queen cells is a day late, can't really take them out, put it there. So you could, instead of buying the expensive tempo queen strips, you could have your own, because you have that 5-year cocktail with 50 queens there. You probably could try that. And that's probably more useful than the other way around.
Your 50 queen cocktail. I did, Marianne Fraser told me once that a nice way to get a new queen introduced is to take the old queen and smash her face against the screen. And like, I can't, because you could get the old queen mandibular pheromone to kind of wear off until the new queen comes in. But that just feels brutal to me. I am not comfortable doing that, but now there are quite a few effective swarm lures, so you could try. I've never tried putting a queen in alcohol, but I did talk to someone recently that said what they do is put some swarm lure in like a Dixie cup or a red Solo cup, and then they just hang that at a very easy place in their yard. And then if their bees do go swarm, hopefully they go and swarm right to that. So then it's really easy to put into the hive, which I thought was very clever, which I haven't tried, but I think that's very clever. All right. Anna, I'm just giving you all the mite questions. I'm assuming you love them. Perfect. So this is from Tim, and it says, since we're in the epicenter of mite concentration, which could we just assume that we all have mites and just skip the testing and treat?
So yes, we should assume that we all have mites. All of our colonies have varroa mites. We should also assume that they do need to be managed, even if the levels are really low. But I think you have much better information as a beekeeper if you're monitoring throughout the season, so you know when those spikes happen, so you can see if your treatments are effective. And also, if you start this early in the season, getting 1 mite in your sample versus 10 versus 30, those are really big differences. So having more information is going to help you, I think, be a better beekeeper and manage mites throughout the season. But that's not, saying that you can do monitoring instead of treatment. Normally, we're assuming that they're in all the colonies and we need to do management throughout the season. And it definitely depends too on how many colonies you have in terms of how much monitoring you're able to do. But especially if you have a small number, or if you're new at beekeeping and don't really know what growth looks like in your area, in your yard, it can be a really helpful way to have more context and information for making those management decisions.
And your mite levels or your mite risk will change every single year. So what you did last year may not work for you this year if one new beekeeper moves into your area or one beekeeper can't manage it. So you want to make sure that whatever you're doing is still working. Dan, so someone made new splits today. Yay. With capped swarm cells. They had 6 frames in a 10-frame box, and then the other 4 are undrawn frames. Should they feed them?
My inclination, based on what I've seen where I'm located in Ingham County, the last week plus would be probably not necessary. Now again, but what are you observing? If your colonies are, I would assume this is a reproductive swarm that created these cells, probably not a crowding swarm. Sorry, I don't know if it's picking up. But I would say that where I'm at, there's an abundance of nectar on the landscape right now. We've had favorable weather for the bees to go get it. So if you're in a similar position and you're, got autumn olive, or there's a lot of stuff out there right now that's going. I wouldn't worry about the feeding at this point. If we had a forecast of 10 days of 52 degrees and raining, then probably wouldn't hurt.
Okay, yes, I agree. And I was also super distracted watching your puppy in the background. Dear friend Margaret says, we split our 2 hives 2 weeks ago and left lots of room, but still captured 2 swarms. A friend on the East Coast had multiple swarms in a week. Is this a particularly swarmy year? So I had 4 swarms in an hour yesterday and another call tonight. And I don't know if it's a particularly swarmy year, but where we all are, we had lots of cold and rain and then one big worm day. And that's kind of when it all happens. I will say that I have definitely had swarms show up in my yard that are absolutely not from my bees. And I know that because I mark all my queens, and when I see a swarm show up, I'll go out and look through my hives and it's not from the bees in that yard. So sometimes they do just show up in your yard. We did have a very, at least, where I'm in Jackson County, and we had cold, warm, cold, warm, cold, warm, and that does allow them to get really broody really early. So I think it was a little bit more dramatic, but I also feel like every year is very dramatic at this time of year. So I don't know if anyone else wants to add to that one. All right, and there is a question. So the next one, I'm going to see if we're losing people because I know we're getting a little bit late, but we've got, I think, 3 more questions here. So someone did ask, are there any vets in the Detroit metro area from whom I can get medicine for European foulbrood? So there is one place you can check now and one place you'll be able to check soon, and then one place that I can help you with. So the Honeybee Veterinary Consortium maintains a website that has a listing of veterinarians who are willing to work with honeybees. So that's the Honeybee Veterinary Medicine Consortium. Wait, Honeybee Veterinary Consortium, HBVC. The Department of Agriculture and Rural Development in Michigan, MDARD, is also going to be hosting a page, just like they do for poultry medicine, of all the vets that are willing to take on veterinary clients. So we're working on that. We've gotten to the point where we have a bunch of vets who are listed. We have 24 that are willing to take vets within, or take beekeeping clients within Michigan. We're just like finalizing and fixing errors on that. So that should be out soon. And so there will be a new resource. We'll let beekeepers know when that comes out. The third option is that tomorrow or Wednesday night, we are hosting a call for all the veterinarians who are willing to take beekeeping clients. And if you follow up with us, we can check to see if anyone will come to the Detroit area. So hopefully we'll have a listing out, but check. Anna just put the link in the chat for the vet consortium. And if you don't hear from anybody, follow up and we can send it to our list of vets in Michigan. And then the next two I have opinions on, but if anyone else wants to follow up too. So Isaac put about an article that they read expressing changes to hive design. And we could put the link in the chat. And it says, do you know if anyone in the broader community is following up with a different style based on this? It's very interesting, especially for us in the North. So the article it came out last year, and it's basically just a look into hive insulation type. So I just put it in the chat. It's open access. And so I don't think anyone's doing dramatic changes to hive design at the moment based on that. So it's kind of like a deep dive into the fact that wood is poorly insulated. And it looks into kind of a lot of the mathematics behind heat loss in a colony. The 2 points that I have in response to that is, one is there's something very beautiful in terms of having just kind of a standard hive design. When I went to Sweden for a year and worked with beekeepers there, where everybody just does their own hive, it's really hard to buy nukes. It's really hard to buy extraction equipment. It's really hard to buy spare equipment. So there is something lovely about having something standardized. Before these last few years, this hive design worked really, really well, or we had very low losses, right? And so we've been using the same hive design for well over 100 years, including periods where we had very high losses or low losses. So the current issues that we have with keeping bees alive are likely not due to hive design. I said that in about the most backwards way possible, but I'm hoping the point got across. But it's not the hive's fault. And we've done some studies both in Michigan and Wisconsin where they've actually overwintered colonies in basically just screens. So you can overwinter a honeybee colony in a screened box with no hive walls, and the colonies can still live. And so what we like to talk about here is, I mean, I've had colonies in cardboard boxes, and, you know, the hive is fine. The hive is the house that it's in. I'm way more interested in your animal being healthy. If you have a healthy animal with lots of food, the hive design doesn't matter quite as much. But it is also like same thing with new queens. It's interesting to try out. And I'm way more excited about what Dan has been working on, or is looking at, like indoor wintering and things like that, is where we can actually control the environment outside the hive. I don't know if anyone else wants to chime in on that. All right. And then the last one, it says, is the Demarest method for splits effective if you do not want to increase the number of hives in your apiary? I know I tried this last year, and where we are in Michigan, that well, it depends on your area. So where I am in Jackson County in Michigan, my nectar flow turns out really hard. I did a classic Demarest on a bunch of hives last year, and they ended up being monsters. And so I basically had to do something again to manage swarm control. Plus, I started getting concerned about mites. So depending where you are and how strong your nectar flow is and how big your colonies are, you could try a classic Demarest and try to recombine them, and it can work. I wouldn't say it necessarily works in all areas. So where I was last year, it was not sufficient. What I ended up going back into was a modified Demarest, where I split the hive like I was going to do a Demarest, and I ended up removing the brood and using it for other purposes. And I can talk about that for an hour and a half. So I'm going to give you the short answer for that. All right, and then the very last question is how to administer European foulbrood medications. And there are resources on that. There's, if you look to, I'll put the link in the chat, but it's Animal Drugs at FDA, and it gives you the 3 methods for administering terramycin, but you should be getting those directions on the label of the drug that you have purchased. So if you're getting terramycin for European foulbrood, it should come with directions, and generally it's applying the dust for that. All right, so I was just decided to hammer out the rest of the questions myself, but those are all my favorite topics of EFB and queen things and demo rays anyway. So thank you everyone for all of your attention. 8:42, which is fantastic that we have so many people that are still interested, and we really appreciate you taking the time of your evening to work with us and learn more about your honeybees.
Great. Thanks, everyone. If you have questions, you can send them through Ask Extension on our website, and we'll see you next month.
Good night.